Hey everybody, in the last couple of videos we have been talking about double strand break repair by homologous recombination. In this video we are going to take a look at a couple other mechanisms that can repair double strand breaks. So specifically we will talk about, I will talk about synthesis dependent strand annealing. So that's a mechanism that can repair a double strand break in a DNA molecule. We'll also talk about single strand annealing, and break induced replication. So these are three different mechanisms, three other mechanisms that can repair double strand breaks in DNA. So first, let's take a look at synthesis dependent strand annealing. Let's see how this works. So this is thought to be the main way a double strand break is repaired outside of meiosis. So let me diagram some DNA molecules here. So this is going to start a lot like double strand break repair, or the DSBR bihomologous recombination mechanism. I don't know why that color is so light. So okay, and I want to add one more color here. So I'm going to add this up. That's too light. Let's see, we need a darker green. We've got all these colors here, but no dark green. Let's see. Okay, maybe this one's a little darker. So I'm going to indicate, a few base pairs in these two strands with this green color right here. Now, this will be the recipient, this will be the donor. The recipient is going to receive information from the donor. It's also going to receive the double strand break. And let's assume that double strand break eliminates, these few base pairs in here. Just obliterates that region right here. So we're left with a recipient that looks like this. Now, I guess I can put the donor down here. The donor is not broken, it's just, I want to keep those bases green colored. So, now, eventually we'll see a mechanism that, just would take these loose ends and stick them together. But that would be a problem, right? Because we lost these base pairs, these green base pairs. And if we did that, then the repair of the double strand break would result in loss of genetic information. So it's in the cell's best interest, when it's repairing a double strand break, to use a donor to repair that break, so it can sort of recoup some of that genetic information that was lost during the break. So in most cases, using a template is the best way to repair the DNA molecule. Otherwise, if the cell stuck these back together, well, those green bases would have been lost. And if this was in the middle of a gene, that could be problematic. So how does this process work? Essentially, we're going to have 5' end resection. So just like in the DSBR model, and then we're going to have a single end invasion, just like in the DSBR model. This is going to invade the donor, and that homology search is going to take place. Let me get a new piece of paper for this. So let me quickly diagram that. Okay. So we have the displacement loop. Oh, and I should mention, make sure you check out the lecture on DSBR by homologous recombination, which is lecture 17, right? Because if you haven't seen this yet, then none of this is going to make sense. So this is the displacement loop. This is the 3' end invasion, the single end invasion. Let's say it found homology here. Now, what can happen? Oh, you know, I forgot to put those green bases in here. Let's see. Okay, let's say it found homology and it's just before that green region that was lost in the recipient. Now, what's going to happen? So that 3' end can be used to synthesize DNA. And now with the DSBR model, what happens next? Well, we have the second end invasion, second single end invasion step, where this will anneal with the displacement loop and start extending this end. However, in this model, in the synthesis dependent strand annealing model, what happens is this right here keeps synthesizing DNA. Eventually, this end keeps being extended. Eventually, it jumps back up, anneals with this one up here because it's complementary, right? Because this strand is right here. This top strand is essentially the same thing as this one down here. So it keeps. So we have DNA synthesis here. Eventually, it jumps up here. That process is poorly understood how that happens, and when that jumps up there, we're going to end up with something like this. Let me see here. And we have that green. So remember, we copied over that part, that green part. So I'm going to color that green. And then you know what? I'll use blue again for this other part here. Okay, so that's annealed here, that's double-stranded there. So now we're left with these gaps, right? And a big gap here. And these are just filled in, right? Because this is a 3' end here and a 3' end here. Those are filled in by polymerases, and that little nick that's going to be left at the end is going to be sealed by DNA ligase. So after all is said and done, the recipient now looks like this. So essentially identical to how it looked at the beginning of this process, right? So here it was before the double strand break. The double strand break came in, blew away all those green bases, obliterated those. We get 5' end resection. We got a 3' end here that invades the donor DNA. It finds homology, synthesizes DNA, and instead of getting the second end invasion and extension, what we get is this jumps up, anneals with this end right here. And then we have these gaps, and the gaps are filled in by polymerases. So, and the nicks, the last nick that would be left right here and right here will be sealed by DNA ligase. So that's the synthesis-dependent strand annealing mechanism for fixing a double strand break. Now let's take a look at the single strand annealing mechanism. So this is SSA, the SSA mechanism, and this is thought to work when there's a double strand break in a region that has a direct repeat. So what is a direct repeat? Well, essentially a direct repeat, let me see, I could take, why don't I take a sequence like this. So here's a sequence of DNA, and I won't put the complementary sequence down here. Let's just look at this sequence right here. And it goes from the 5' to 3' direction. And let's say there's some random bases in here. And somewhere downstream, we see the exact same sequence again. So this would be called a direct repeat. And there's no indirect repeat. The opposite of a direct repeat is an inverted repeat. So this sequence would be inverted down here somewhere. But a direct repeat is when you have the same sequence repeated one after another in the same orientation on a strand of DNA. So we have some direct repeats in the human genome. We've covered some of those. One of those would be the ribosomal DNA arrays, right? We have our DNA genes repeated one after another on the acrocentric, on the P arms of the acrocentric chromosomes, 13, 14, 15, 21, 22. So this single strand annealing step mechanism is thought to work when we have a double strand break in a region that contains direct repeats, or it can work. It doesn't always have to be the one that is used. So how does this work? Well, single strand annealing, the SSA mechanism, I'm going to diagram it with two colors, and we don't need to look at a recipient DNA molecule and a donor, because the recipient isn't receiving information from a donor. So there's no recipient, no donor in this mechanism. So I'm using green to indicate the location of the directly repeated sequence. So, and let's say the double strand break occurs in here. So if it occurs in here, well, okay, so the first step of this mechanism is going to be similar to the others. We're going to see 5' end resection. So that's going to give us something like this. So right, these are the two strands of the DNA molecule. Double strand break broke both strands. 5' end resection started here, chewed all the way down to here. So now, so one of the different things about this mechanism here, there's no strand invasion of a donor. And why is that? Well, because we have homology between these two. These direct repeats are, direct repeats. They're identical. So, or not identical, but complementary. So what's going to happen, or what can happen in this mechanism, is that these are going to anneal. And we're going to get these little flaps right here. Because this blue, these probably aren't complementary. Or don't have to be complementary. So this little blue piece right here is going to be a flap. And why is it a flat? Because it doesn't anneal with this piece. It's not complementary to that. Those aren't complementary. Same thing over here. Okay, so let's put this here. Okay, so what are we left with now? These have to be trimmed away by some exonucleases. Or maybe we have some endonucleases involved, trimming down here somewhere or cutting down here somewhere. I'm not really sure. I don't know if that part of the model has been worked out yet. After those are removed, we'll be left with a gap, right? Gap here, and anytime we see a gap in a DNA molecule, we can just assume that the 3' ends are used by a DNA polymerase. We don't need to worry about which DNA polymerase in a eukaryotic cell is going to do the extension there. And then the nicks will be sealed by ligase. So that's easy. Essentially, anytime we see a gap, we know what's going to happen. DNA polymerase is going to come in. It's going to extend the 3' end, which is going to be right here after that piece is trimmed away, and right here after this piece is trimmed away. Extend those. The last little nick that remains will be sealed by DNA ligase. Now, you may have noticed that this is not the best way to repair a double strand break, because what happened? We went from directly repeated sequence to having just a single iteration of that sequence, right? So we lost some genetic information when this mechanism was used to repair the double strand break. And it's thought that the SSA mechanism is probably a cause of some genetic diseases. One of them would be the alpha thalassemia. So where we don't have enough copies of the alpha globin genes. And how could that be the case? Well, remember that alpha 1 and alpha 2, well, those are nearly identical. So those are essentially direct repeats. So we can have the SSA mechanism repairing a double strand break in the alpha 1 alpha globin gene cluster, and that can lead to loss of one of these genes. So we also talked about earlier how unequal recombination could lead to loss of one of these guys too. So we have a couple different processes that can lead to alpha thalassemias and other genetic diseases that where there's more than one copy of a gene or paralogous genes, and you end up losing one of the genes because of the SSA mechanism of repair of the double strand break. So, okay, let's take a look at one more. Break-induced replication. Or break-induced replication repair, often abbreviated as BIR. Now, break-induced replication repair, so how does that work? So, now there is a donor and a recipient in this repair mechanism. So we start off very similar to the other mechanisms, where we have a double strand break, and what comes next? You should probably know what comes next. 5' end resection. Well, by now we should know what comes next. Now, so this is where the mechanism becomes different. The repair mechanism becomes a bit different from the other mechanisms. So break-induced replication repair is thought to occur when something went wrong. Let's say one of these ends, so this is one end of the double strand break that's been resected. This is the other end. One of them is lost. For some reason, one of them is lost. And when one of the ends is lost, well, what would happen? You know, this is sort of like an emergency repair process. The cell would die without this end of the chromosome. Remember, because this goes to one telomere, this goes to the other telomere. So it's sort of an emergency repair process. And what happens, let's take our donor down here. And the orientation is going to be a little different here, I think, than I usually put the 3' strand up top, but we're going to see why I'm going to do it this way. I usually put the 3 on top and the 5 on the bottom, but you'll see why I'm doing it differently this time. So 3, 5. Okay, so what happens is this has been lost. So this now is the only remaining end that the cell has to work with. It's going to be used for the homology search. Ideally, it finds a match, and we end up with something that looks like this. So let me put the D loop, displacement loop in the donor. And the single end invasion, found homology right here. Okay, so found homology. Now what can happen? That can be extended. But remember, there's no other end up here. There's no way that this can stop and just anneal with the other end. And we can continue with, say, double strand break repair by homologous recombination. That can't happen here because we lost the other end. The cell lost the other end. So what happens is this displacement loop will move all along the donor template, and the polymerases will keep extending all the way to the end of the donor template. And so at the end, what we have, well, once it reaches the end, what we have, this has been extended, so eventually it dissociates from the donor template. And the donor just goes back to how it was before this process started. Okay, now we have one long single-stranded piece of DNA right here. So essentially what this needs is essentially one long gap right here. So you can probably imagine what needs to happen, right? DNA polymerases need to come in. We're going to need primers here. So this is a lot like DNA replication during S phase. So this whole piece is going to need to be, this whole single strand is going to be needed to be made. And we don't need to worry about the details of how that happens. Okay, so that's essentially break-induced replication. Let me put, I'll fill this in here. We'll say missing DNA filled in by polymerase. Okay, so that is break-induced replication repair. In break-induced replication repair, it's kind of, it's interesting, right? Because it results in how do we know that break-induced replication repair exists? Well, before working out, like, that model probably exists, and I'm actually not sure about the experiments that have been done, but this can happen sometimes. One of these days I'll actually read some research on people who study break-induced replication repair. But let's take a pair of homologous chromosomes in a random organism. You could even say a human. And let's say this is chromosome 1 from this individual's father, and this is chromosome 1 from the individual's mother. And I'll color this one in blue. I'll color this one in red. Now, okay, let's assume a double strand break happened somewhere in here. And after the double strand break, one of the ends, one of the DNA ends was lost. I'll put it over here. Say the cell can't find it. So now when this chromosome over here is used as the donor to fix this over here, what's going to happen? Well, the result essentially is that whatever sequences were over here are now copied over to the other chromosome. So let's say on somewhere up here on this chromosome 1, let's say there are hundreds of genes. Let's say one of them has the big A allele for a certain gene here, and over here, one of them over here the gene has the small a allele. So there's a gene A up here in this segment. Gene A, this individual inherited the big A allele from the father and the small a allele from the mother, but because of break-induced replication and where the double strand break was and which part of the chromosome was lost, well, what happens is, that small a allele and every other gene that was over there, all the alleles that were found for the genes over here are now copied over here. So the genotype of this cell changes after the repair process. So now, instead of being heterozygous, big A, small a, it now has two little a's. And the same thing happens for every gene on this end of the chromosome. So that's break-induced replication repair, and that's one of the things that can happen when break-induced replication repair is used to repair a double strand break. You can go from a heterozygous condition for many genes to the homozygous condition for those same genes after the repair process. Okay, I wanted to touch on one more thing in this video before we talk about gene conversion. So going back to double strand break repair by homologous recombination. Now remember, we had this step of the mechanism that produced a double holiday junction. So this is the double holiday junction that results during double strand break repair. And in order to resolve this structure, we need to make some breaks, right? So that these strands can be cut. Oh, sorry. This blue one and this red one right here, or this blue and this red one right here. Or we can cut the green and the red, or the blue and the red. So these actually need to be cut. You can't just pull these apart. Another thing that can happen is that we can call these branch points. Branch points or junctions. Let's just call them junctions. A lot of people call them branches. So we can have migration of these junctions or branches. Let's see, we're going to call this branch. It's officially called branch migration. So the junctions are called branches. So these junctions can move. So this can actually move that way, and this one can move that way. Or they can come closer together. They can't eventually merge and then the situation be resolved because they're still tangled a little bit, but the branches can move. And that's going to be important for this process called gene conversion. So the further apart the branches move from one another, the more these DNA sequences are affected by this process called gene conversion, which we're going to look at in the next video. So one of the things I wanted you to see or think about is, why is it so hard to resolve a double Holliday junction? Why is it so hard to resolve? Well, part of the reason why it's hard to see why it's hard to resolve is because of the way we traditionally diagram these things with just lines on top of one another. But when you think about it, this is DNA, and DNA is a double helix, right? So these strands are wound around each other. So it would look, this is probably a better figure or better diagram right here. So this is also the double Holliday junction, and it probably represents what double Holliday junctions really look like in the cell. We should probably have some nucleosomes in here too, but we can see how the strands are wrapped around each other, and then we have one of the strands coming down here, and now it's wrapped around the donor template right here. We have this green wrapped around with the red from the donor, and then we have it moving back up here again. So you can see how you can't just pull these things apart. The cell actually needs to cut the strands and ligate them back together to resolve the double Holliday junction. Also, this might be easier to see how these can move now. So if this should move this way, essentially what you have is this being wrapped around these guys down here, and this one going up here being wrapped with forming a helix with this guy up here. So you see a lot of like spinning of the DNA molecules or the helices as these branches are moving. Okay, well, I hope at least that this helps you see why it is difficult to resolve the double Holliday junction and why we need some endonucleases to come in here and cut the strands in certain places to resolve this structure. Okay, in the next video I will talk about gene conversion.